Welcome, everyone. Today we're going to embark on an adventure into the world of scientific papers and data analysis. Now, I know what you're thinking. Scientific papers sound about as exciting as watching paint dry. But trust me, by the end of this lecture, you'll see that writing a scientific paper is like crafting the ultimate detective story, where you're both the detective and the storyteller. Science is fundamentally about asking questions and finding answers. Every scientific paper follows this exact journey, from curiosity to discovery. Let's start by understanding the basic structure. A scientific paper has 6 main sections, and each one has a specific purpose. Think of it like building a house. You need a strong foundation, walls, a roof, and all the parts must fit together perfectly. Here are the 6 sections stacked like building blocks. The title is your first impression. The abstract is your elevator pitch. Introduction sets the stage. Methods explains how you did it. Results shows what you found. And discussion explains what it means. Let me ask you a question. Why do scientists bother writing papers at all? Why not just do the experiments and keep the results to themselves? The answer is that science is a conversation. Imagine if Isaac Newton discovered gravity but never told anyone. We'd all still be confused about why apples fall down. Scientific papers let us share discoveries, build on each other's work, and push humanity forward together. Let's dive into the first section, the title. Your title is like a billboard on a highway. You have about 3 seconds to grab someone's attention before they drive past. A good title is specific, informative, and intriguing. Compare these 2 titles. The bad one is vague. What about plants? Are we studying their feelings? The good one is specific. It tells you the variable being tested, blue light, the organism, tomatoes, and the measurement, growth rate. Big difference. Imagine if famous papers had terrible titles. Instead of On the Origin of Species, Darwin could have written some thoughts about animals and stuff. Instead of A Mathematical Theory of Communication, Shannon could have called it How to Send Messages, I guess. See why specificity matters. Next up is the abstract. This is arguably the most important section because most people will only read your abstract. You have about 250 words to summarize your entire study. That's roughly the length of a text message conversation with your friend about weekend plans, but you need to fit your entire scientific discovery in there. A good abstract has 4 components: background context. Why should anyone care? Methods. What did you actually do? Results. What did you find? And conclusion. What does it mean? Think of it like a movie trailer. Give away just enough to make people want to read more, but not so much that they don't need to. Let's do a quick calculation. The average person reads about 200 words per minute. Your abstract is 250 words. That means someone will spend about 75 seconds reading your abstract. In those 75 seconds, you need to convince them your work matters. No pressure, right? The introduction is where you build your case. Think of it like a lawyer's opening statement. You need to convince the jury, your readers, that your research question is worth investigating. A great introduction follows a funnel structure. Start broad, then narrow down to your specific question. Start at the top with the big picture. Then zoom in on what other scientists have discovered. Identify the gap. What don't we know yet? Finally, present your specific research question. It's like zooming in with a camera from a wide shot to a close-up. Let's say you're studying the optimal cheese distribution on pizza. Your intro might go, broad context. Pizza is consumed by billions worldwide. Previous research, studies show even cheese distribution affects consumer satisfaction. Gap. No one has quantified the perfect cheese-to-crust ratio. Your question. Does a mathematical model exist for optimal cheese placement? See how we funnel down? Here's a question for you. How long should your introduction be? Should it be 1 paragraph or 10 pages? The answer is roughly 10 to 15% of your total paper length. For a typical research paper of 3,000 words, that's about 300 to 450 words. Long enough to make your case, short enough to keep people reading. Remember, you're writing a research paper, not a novel. Now we get to the methods section. This is your recipe. If science is cooking, then the methods section is where you write down exactly how you made your dish so that someone else could recreate it perfectly. The golden rule is reproducibility. Imagine handing your methods section to a scientist on the other side of the world. They should be able to follow your instructions like an IKEA manual, hopefully with better results than most IKEA furniture, and get the same results you did. That's reproducibility. You need to include specific details. Don't just say, "We heated the solution." Say, "We heated the solution to 95 degrees Celsius for 30 minutes, using a hot plate set to medium-high." Don't say, "We measured the plants." Say, "We measured plant height from soil surface to apical meristem, using a digital caliper accurate to 0.1 millimeters. Your methods section needs 3 key components. First, describe your sample. What are you studying? Second, list your equipment and materials. Third, explain your procedure step by step. Think of it as 3 legs of a stool. Remove one and everything falls apart. Imagine if a chef wrote methods like some scientists do. Bad methods would say, "Make a sandwich." Good methods would say, "Place 2 slices of whole wheat bread, each 1 centimeter thick, on a clean cutting board. Apply 15 grams of peanut butter to 1 slice using a butter knife in smooth, even strokes. Add 20 grams of grape jelly to the second slice. Combine slices with spread sides facing inward and apply 50 newtons of pressure for 2 seconds. Now that's reproducible." The results section is where you present your data. And here's the crucial part. Just the data. No interpretation, no opinions. No, "I think this happened because." You're a journalist reporting facts, not an opinion columnist. Save your interpretations for the discussion section. Present your data using graphs, tables, and figures. A picture is worth 1,000 words, and in scientific papers, a good graph can save you from writing 3 paragraphs of dense numerical descriptions. Make your graphs clear, label everything, and use error bars to show uncertainty. Rule 1. Every figure needs a descriptive caption. Rule 2. Every table needs column headers that make sense. Rule 3. If someone looked at your graph without reading anything else, they should understand what you measured and what you found. Your figure should tell the story, even if someone just flips through looking at pictures. Here's an important question. Should you include all your data, or just the important parts? The answer is to include your most important and representative data in the main paper. If you collected 500 measurements, you don't need 500 graphs. Show the key findings and mention the rest in text. You can put extra data in supplementary materials. Think quality over quantity. Show me your greatest hits, not every song you ever recorded. Now let's talk about the backbone of good science, data analysis and statistics. This is where many students start sweating, but don't worry. Statistics is just a tool to help us separate signal from noise, real patterns from random chance. Statistics helps us answer a fundamental question. Is what I'm seeing a real effect or just random variation? If you flip a coin 10 times and get 6 heads, is the coin biased or did you just get lucky? Statistics gives us tools to make that call. One of the most important concepts in statistics is the p-value. The p-value tells you, if there was actually no real effect, what's the probability I'd see results this extreme just by random chance? Scientists typically use p less than 0.05 as their threshold. That means less than 5% chance of seeing this by accident. When your p-value is less than 0.05, you can say your result is statistically significant. That's science speak for this probably didn't happen by accident. When p is greater than 0.05, you can't confidently say there's a real effect. The line at 0.05 is like the border between probably real and could be random. Let me introduce you to the 3 most common statistical tests high school students encounter. First is the t-test, which compares the means of 2 groups. Did the experimental group really differ from the control group? Second is ANOVA, which compares 3 or more groups. Third is correlation analysis, which asks if 2 variables are related. Let's do another quick calculation. How many samples do you need for a good experiment? Here's a rough rule of thumb. For detecting a medium-sized effect with 80% confidence, you need about 30 samples per group. So if you're comparing 2 groups, that's 60 total samples. Comparing 3 groups, you need about 90 samples. More samples mean more confidence in your results. Here's the relationship between sample size and confidence. 5 samples, your results are shaky. 30 samples, now we're talking. 100 samples, you've got rock-solid data. More samples reduce uncertainty and increase statistical power. In statistics, there are two types of errors you can make, and they have the least creative names ever: Type 1 and Type 2 errors. But understanding them is crucial for interpreting results correctly. Type 1 error is a false positive, like a fire alarm going off when there's no fire. You concluded there's an effect when actually there isn't. Type 2 error is a false negative, like a smoke detector failing to go off during a real fire. You concluded there's no effect when actually there is one. Imagine you're testing if a new cookie recipe tastes better than the old one. Type 1 error. You conclude the new recipe is better, you switch production, turns out people hate it and you lose customers. Type 2 error. You conclude the recipes taste the same, you stick with the old one, turns out the new recipe was amazing, and your competitor steals it and becomes rich. Both errors have consequences. So here's a philosophical question. Can we ever be 100% certain about a scientific conclusion? The honest answer is no. Science deals with probabilities, not absolute certainties. Even with p less than 0.05, there's still a 5% chance you're wrong. That's why we replicate studies, why we demand peer review, and why science is self-correcting over time. We're always refining our understanding. Finally, we reach the discussion section. This is where you get to interpret your results, explain what they mean, and put them in context. Think of it as the "so what" section. You've shown your data. Now explain why anyone should care. The discussion connects your specific results to the bigger picture. Start by summarizing your main findings in plain English. Then explain how they fit with previous research. Do they confirm what others found? Contradict it? Fill in a gap? Finally, discuss the implications and suggest future research directions. A strong discussion has 4 components. First, restate your key findings without just copying the results section. Interpret them. Second, compare with previous studies. How does your work fit in? Third, acknowledge limitations. No study is perfect. Fourth, suggest future directions. What should the next scientist study? Here's something students often forget. Acknowledging limitations makes your paper stronger, not weaker. Every experiment has limitations. Maybe your sample size was small. Maybe you couldn't control some variable perfectly. Maybe your equipment had limitations. Being honest about these shows you understand the scientific method. Reviewers respect honesty. When you hide limitations, reviewers will find them and question your credibility. When you openly discuss them, you show scientific maturity and understanding. It's the difference between pretending you're perfect and being a genuine, thoughtful scientist. Let's spend some time on one of the most powerful tools in science communication: graphs and data visualization. A well-designed graph can make your argument instantly clear. A poorly designed graph can make brilliant research look confusing. Here's a good graph. Clear axis with labels and units. Data points marked clearly. Error bars showing uncertainty. A legend if you have multiple data sets. Clean and uncluttered. Someone should be able to understand your key finding in about 5 seconds of looking at this graph. Let me tell you about common graphing mistakes. Mistake 1. Axes without labels or units. I've seen graphs where the y-axis just says height, in millimeters, meters, kilometers, who knows? Mistake 2. Misleading scales that exaggerate tiny differences. Mistake 3. Pie charts when a bar graph would be clearer. Mistake 4. 3D graphs that look cool but are actually harder to read. Want to know a secret? The human brain is amazing at comparing things visually, but terrible at comparing lists of numbers. Would you rather read, Group A had a mean of 23.4 with standard deviation 3.2, while Group B had a mean of 31.7 with standard deviation 4.1, or look at a bar chart showing Group B is clearly higher? Visual comparison wins every time. Look at this bar chart. Instantly, you can see Group B has a higher value than Group A. No calculations needed, no squinting at numbers. This is the power of good data visualization. It makes your point obvious. Let's talk about error bars. These are those little lines sticking out of your data points. And they're incredibly important because they show the uncertainty in your measurements. Science is never perfectly precise. Every measurement has some uncertainty. Here's a data point with error bars. The point shows the average value, and the error bar shows the range of uncertainty. Maybe it's standard deviation, maybe it's standard error, maybe it's 95% confidence interval. Just make sure you specify in your figure caption. Let's do a quick uncertainty calculation. Say you measure a plant's height 5 times and get 10.2, 10.5, 10.1, 10.4, 10.3 centimeters. The average is 10.3 centimeters. The standard deviation is about 0.15 centimeters. So you'd report your measurement as 10.3 plus or minus 0.15 centimeters. That plus or minus part, that's your error bar. Let me give you a real-world example of how all these concepts come together. Think about climate science and measuring global temperature change. Scientists measure temperature at thousands of stations around the world. Each measurement has uncertainty. Is the thermometer perfectly calibrated? Is it in the right location? They use statistics to combine all these measurements and calculate a global average with error bars. This is data analysis in action on a planetary scale. This shows the scientific method beautifully. Question: is Earth warming? Hypothesis: yes, due to greenhouse gases. Methods: measure temperatures globally over decades. Results: average temperature has increased about 1 degree Celsius since 1880. Discussion: this matches predictions from physics and has major implications. That's a complete scientific paper in miniature. Before we wrap up, let's talk about something crucial: ethics. Science depends on trust. When you publish results, other scientists trust you reported honestly. That trust is the foundation of all scientific progress. There are 3 major ethical violations in science: fabrication, where you make up data that doesn't exist; falsification, where you manipulate real data to show what you want; and plagiarism, where you steal someone else's work. All 3 are scientific crimes that can end careers. Don't do them. There's also a subtle form of dishonesty called cherry-picking. This is when you only report the data that supports your hypothesis and hide the data that doesn't. Maybe you did the experiment 10 times, it worked twice, and you only report those 2 times. That's cherry-picking, and it's wrong even if technically the data you reported is real. Students often ask, what if my data doesn't support my hypothesis? Should I redo the experiment until it works? Here's the truth. Negative results are still valuable results. If your hypothesis was wrong, that's an important finding. Maybe your hypothesis needs refinement. Maybe there's no effect. Maybe you discovered something unexpected. Science advances just as much from finding out what doesn't work as what does. Report your data honestly, whatever it shows. Let me give you some practical tips for writing your first scientific paper. These are lessons I've learned from reading hundreds of student papers. Tip 1. Start with an outline. Don't just start writing from the title and work your way down. Plan out each section first. What points do you need to make? What data supports each point? An outline is like a roadmap. It keeps you from getting lost in the middle of writing. Tip 2. Write the methods section first. It's the easiest section because you're just describing what you did. Getting this done gives you momentum and confidence. Plus, writing methods helps you remember details you might forget later. Tip 3. Make all your figures and tables before writing the results section. Once you have your visual data representations, writing the text that describes them becomes much easier. Let the figures tell the story, then add words to support them. Tip 4. Write the discussion section while your results are still fresh in your mind. Don't wait weeks after finishing data collection. Your insights about what the data means are clearest right after you analyze it. Tip 5. Edit ruthlessly. Your first draft will be too long and probably confusing. Cut unnecessary words. Simplify complex sentences. Remove redundancy. Good scientific writing is clear and concise. If you can say it in 10 words instead of 20, use 10. Here's an example of editing. The first version is wordy and redundant. Obtained results indicate clearly. That's 5 words. That could be 2. The edited version is cleaner and stronger. Same meaning, half the words. That's good scientific writing. Let me share some common writing mistakes I see in student papers so you can avoid them. Mistake 1. Using first person too much. Instead of, I think this shows, or we believe that, use, the data suggests, or these results indicate. Let the data speak, not your opinions. Scientific writing focuses on evidence, not the writer. Mistake 2. Vague language. Words like many, some, a lot, very are too imprecise for science. Instead of temperature increased a lot, say temperature increased 15 degrees Celsius. Give specific numbers whenever possible. Mistake 3. Confusing correlation with causation. Just because two things happen together doesn't mean one caused the other. Ice cream sales and drowning rates both increase in summer. Does ice cream cause drowning? No, they're both caused by a third factor, hot weather. Be careful with causal language unless you have experimental evidence. Finally, let's talk about what happens after you write your paper. In the real world of science, papers go through peer review. Other scientists read your paper critically and decide if it's good enough to publish. Here's the process. You submit your paper to a journal. The editor sends it to peer reviewers, other scientists in your field. They read it critically, check your methods, verify your statistics, and look for errors. Then, they send feedback. Almost always, you need to revise and resubmit. This cycle might repeat several times before publication. Peer review isn't about rejecting papers. It's about making them better. Reviewers catch errors, suggest improvements, and make sure conclusions are justified by the data. Yes, it can be frustrating when reviewers criticize your work, but they're helping you and helping science be more accurate. Alright, let's wrap up with the key takeaways from this lecture. We've covered a lot of ground, so let me distill it down to the essentials. First, scientific papers tell a story. Every section has a purpose, and they all fit together to take the reader on a journey from, here's an interesting question, to, here's what we found and what it means. Second, reproducibility is sacred. If another scientist can't repeat your work, it's not science. Write your methods clearly and completely. Third, statistics are your friend, not your enemy. They help you figure out if your results are real or just random chance. Learn the basics and use them. Fourth, good visualization is worth its weight in gold. A clear graph can communicate your findings faster and better than paragraphs of text. Fifth, ethics aren't optional. Report your data honestly, acknowledge limitations, give credit where it's due. Science depends on trust, and trust depends on integrity. Sixth, negative results matter. Not every hypothesis is correct, and that's okay. Finding out what doesn't work is still advancing knowledge. Remember, science is a cycle. Questions lead to methods. Methods produce answers, and those answers generate new questions. Writing a scientific paper is documenting one turn of this eternal wheel of discovery. So what should you do next if you want to write your first scientific paper? Step 1. Pick a question you're genuinely curious about. It should be specific enough to investigate in a reasonable time frame. Don't try to solve world hunger in your first paper. Maybe just figure out which fertilizer makes tomatoes grow fastest. Step 2. Read papers. Find 5 to 10 papers related to your topic. See how real scientists structure their arguments, present data, and write discussions. You'll learn the style and conventions of scientific writing by seeing examples. Step 3. Design a simple experiment with proper controls and enough replication. Remember our discussion about sample sizes? Aim for at least 30 samples if possible. Step 4. Keep detailed notes while you conduct your experiment. Future you will thank present you for writing down exactly what you did. Trust me on this. You'll forget details faster than you think. Step 5. Analyze your data carefully. Calculate means, standard deviations, run appropriate statistical tests. Create graphs that clearly show your findings. Step 6. Write your paper following the structure we discussed. Then revise it. Then get feedback from a teacher or mentor. Then revise it again. Good writing is rewriting. Let me leave you with one final thought. Yes, writing scientific papers involves a lot of technical skills. Statistics, graphing, precise language. But at its heart, science is about the joy of discovery. It's about asking, what if, and why, and how, and then finding out. Every scientific paper is like sending out an explorer into the unknown. You start with a question, venture into unexplored territory, and bring back new knowledge to share with the world. That's incredibly exciting. When you write a scientific paper, you're joining a conversation that's been going on for centuries. You're standing on the shoulders of giants like Newton, Darwin, Curie, Einstein, and countless others. You're adding your voice to humanity's collective understanding of the universe. How cool is that? Remember, science is a team sport. Yes, you might work on your paper individually, but you're building on others' work, and others will build on yours. Your paper isn't the end of the story. It's a chapter in an ongoing epic that spans all of human history. So here's my challenge to you. Go out and write a scientific paper. Pick something you're curious about. It doesn't have to cure cancer or solve climate change. Maybe test which brand of batteries lasts longest. Maybe measure how temperature affects yeast fermentation. Maybe investigate if listening to music helps plant growth. Whatever interests you, your scientific paper starts today. The world is full of unanswered questions waiting for curious minds like yours to investigate them. So grab a notebook, start asking questions, and begin your journey as a scientist. Thank you for sticking with me through this lecture. I hope you now see that writing a scientific paper isn't some impossible task. It's a learnable skill that gets easier with practice. Every great scientist started exactly where you are now. You've got this. Now go make some discoveries and share them with the world. Good luck and happy experimenting.